Here is where we stopped last class, right? Talking about PCOA and 2 and 3 dimension plots. So I'm just gonna give you a few more examples because this is really, I guess, the most important way that we use to visualize bacterial community similarities. In this example here, it's a classic study. They compare the oral microbiota of mothers giving birth. And then, so you can see a clustering of their oral microbiota here in green. And then they also analyzed the microbiota of their babies after they were born, comparing to the vaginal and skin microbiota. And what we can see here is that babies born by natural delivery, they had a very similar microbiota to the vaginal cavity of their mothers. And babies that were born by C-section, they were colonized by the bacteria present in the skin of their mother. And further studies show that they also had the bacteria from the hand of the nurses that were helping with the delivery. So it is a very nice way to visualize the clustering of the different types of bacteria in this study. In this other study, Diego, he was evaluating the microbiota of healthy calves comparing to calves with diarrhea. And what we can see here in red, it's a nice clustering, very evident, showing that the fecal bacteria of calves with diarrhea is quite different from healthy animals in the same farm. Another study comparing different types of diet in, I believe, the rumen of cows, a very recent study. And here, again, we can see that cows that were getting forage, hay, they had a very different microbiota compared to cows that had high levels of carbohydrates. And this one was comparing the microbiota from rumen of different species and different diets, different geographic locations, creating a kind of a map of the ruminal microbiota of animals all over the world. So I'm gonna ask you, those plots that you can see here, what type of plot is that?
Relative abundance.
I'm sorry? Who said that?
I did.
Sarah, what did you say? Relative abundance?
Yes.
Yeah, that's correct. So if you look here, it's just another way to visualize. Instead of columns, you have those pie charts that you can compare side by side the relative abundance, and you should always search for the legend. So here you have the bacteria and the archaea with different colors. So probably this bacteria and this one archaea in the rumen of animals in Australia, for example. And when you look at the PCOA of those animals, the rumen of those animals, what you can see is the formation of many different clusterings, but basically you can break that into four, and you can see that this group here is comprised, for example, by goats that were receiving carbohydrates or supplementation with concentrate, okay? And the data really shows that what drives the composition of the microbiota is the species. So here you have cervids and goats and caprines, and here in this other half of this line here, you can see that it's the microbiota of bovine. And then if you draw another line, you can see that all the samples that were on this side of the line, they were animals receiving hay or pasture, and all the animals on this side of the line, they were animals receiving supplementation with concentrate. So two variables that can drive composition here, species and diet. And you can see very evidently that by a PCOA that they cluster completely, they are separated by those two variables. And this is another classic plot of PCOA from Rob Knight's group, in which they show the composition of the different parts of the human body. And I can't remember exactly, but here is feces, here I believe it's skin, or I think pink it's oral microbiota, vaginal microbiota, skin, and fecal. And I guess in this example, they were talking about the microbiota of a person who got a course of antibiotics, and from normal, you can visualize in an animated way how his microbiota shifts completely towards a dysbiotic state, and therefore a different, not normal fecal microbiota, and after time it starts to return back to a normal composition to what it was before. Right, and I guess all of you have seen this plot here, in which we show the changes of microbiota from horses during treatment with TMS, so antibiotic, sulfatrimetoprim. And what we can see here is at baseline, that's not gonna work. Maybe I have to change again. Oh, it's here. Okay. So here is baseline, and then after 5 days of treatment, you can see that the composition of the bacteria changes completely. And then at 14 days, it starts to come back to normal. And 30 days, it's already close to what it was before at baseline. So it's another way to visualize the dynamics of this composition over time in animals treated with antibiotics. I hope PCOAs are clear to everybody. And now we're going to start talking about refraction curves. And as you can see from here, this is just a very basic analysis in which we represent the number of sequences, the number of DNA sequences that we analyzed in the x-axis. And on the y-axis, what we do is the number of whatever you are analyzing. If you're analyzing number of genera, if you're analyzing number of species or OTUs or phyla, whatever type of number of or counting of different organisms, you can represent them here at the y-axis, okay? And as you can see, this is what we find after we plot that. So just basically, let's give you an example here. If I get 200 reads from this analysis, I'm gonna look here and I'm gonna see that this sample, it had about 20 different genera. But with the same number of reads, this other animal here, they had already the double of the richness. So it had around 40 different genera, okay? So in reality, what we are doing here is to evaluate the different number of genera or species, which is a concept of richness, correct? I hope this is clear to everybody. So after we evaluate, in this case, 4,700 something reads, we can see that this first animal here, it had a richness of about 42, 43 different genera, but this one here had a much higher diversity because it had close to 80, okay, different genera. So we are looking at this and evaluating the different number of the difference in richness between those samples. Is that clear to everybody? Now, there is something else that this can tell us. What else besides richness we can look or we can observe in this plot? Any of you can tell me.
Community membership.
No, we don't know anything about membership here because membership is gonna tell us who is there, right? We don't see, we cannot tell which type or which species is there. This is just a counting of bacteria or different types of bacteria and therefore richness, but there is another alpha diversity concept that we can have a rough idea just looking by those plots, which is what?
Diversity.
Diversity, yes, very good. So if you look at, let's take those two border lines here as an example. They do have more or less about the same richness, right? One has, let's say, 42 species and the other one has 43 or genera, different genera, right? But if you look at the shape of their curves, they are quite different, right? One starts to flatten right about 200 or 300 reads, or not flat, but it starts to go down a bit. But this one, it continues to go up, 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 up. Meaning that for every sequence I get from this sample, I find a new genera or species, if you like. Is that clear? So we say that if I increase my sampling effort, I'm still getting new genera. Every time I get a new sequence, I'm getting a new genera. And therefore, the curve is much more steep at the beginning of the plot just because it has a higher diversity. Remember the concept of diversity, or at least of one of the formulas, that the probability that for every sample or every DNA strand that you take out of the bag, you're gonna find a new, a different type of bacteria. That's what's happening right here. I'm getting a new read and I'm finding a new species or a new genera. Okay, so if you look at the shape of the curve, you can tell that the diversity of this sample over here, it's higher than the diversity of this sample over here, but they have the same richness at the end. Good? Has anybody not understood that? All right. So the other thing we can see is that the other thing that a rarefaction curve can tell us is that what we call sampling effort. Okay, the sampling effort tell us if the analysis we did. Wait a second. Okay, tell us if the analysis we did, it was enough, it was sufficient to find all of the species or most of the species present in that environment. Okay, for example, in this study, I think, we found or we used a subsample of 4,700 reads, okay? And my question is, so with this number of reads, I found 42, between 42 and 78 different genera. If I carry on sampling, if I carry on analyzing DNA sequencing of those or DNA strands of those samples, am I going to find more species than that or not? So there is another type of analysis we do that we call coverage analysis. That's gonna tell me with this subsampling, with 4,700 reads, you have found around 99% of all the genera present in that environment. Meaning that if I carry on spending money and analyzing more and more DNA strands, I'm not going to find a whole lot more of genera there. Okay, that's it. It was good. Meaning that if I continue, sorry, if I continue here, this curve is going to be what? It's going to be flat. Okay, it's not going to go up. If I carry on sequencing them, it's not going to go up. It's going to carry on, it's going to continue in a flat shape. And that's what we want. We want a curve that there are more, all of them quite flat and not steep, not going up. Okay? Is that clear to everybody? Good. So this is an example I made up just to show you again the difference between two samples that have the same richness and therefore more or less 110 genera, but they have a different shape. So which one has a higher diversity, the blue or the orange one?
The orange
The
one?
blue.
Yes, good. So the orange has higher diversity because you can see that for every new read I get, and this should be times 10 or times 100, for every new read I get a new or a different genera. Okay, so it has a higher diversity. Now, this is a real life example in which you can see the rumen microbiota of newborn calves or pre-ruminant calves. And who had and here we have samples from adult cows under different diets, and I think they have beef and dairy cows. So what it's quite obvious that the rumen of neonates, they have a much lower richness because they have less different types of bacteria. And the rumen of adult animals, adult bovine, they have a much richer and a much more diverse microbiota, right? Because you can see here for every new sample you get, you find different new OTUs, and therefore they have a much higher diversity. Okay? Now, this one is just an example to show you that this study, I cannot not even remember what they were evaluating, but you can see that for this number of sequences they were evaluating, those samples, those curves are still going up, in a way. So they are very steep, and therefore, if they carry on sampling more and more, they would find many more different OTUs there. There are many different ways to interpret that. For example, one detail of relative abundance plot, or sorry, rarefaction curves plots is that you can stretch the plot, and that's gonna look like the curves are flat. But in reality, they are not. Okay, I have seen that during reviewing process and say, look, you need to calculate the coverage for those samples, because just looking at those curves, you cannot really tell the curves are flattened or if they are still going up. And it's an easy calculation to make, just to make sure that the sampling effort was adequate. Okay. The other thing is that when you see something like that, it depends of the environment. If you are talking about feces, that's fine. You're probably gonna have a very high richness and diversity of those samples, or soil, for example. But if you are talking about other types of less contaminated environment, what this a plot like that would make me think that the bioinformatic analysis was not very straight, or not very strict, I should say, in a way that many reads, instead of being clustering together because they belong to the same species, they were split in two, and therefore you classify them as two different species, or even if you're not classifying them yet, if you're working at the OTU analysis, then those two reads that eventually could be clustered together because they're very similar, they are still behaving as two different types of reads, and therefore they are classified as two different OTUs. And here you see a huge richness. Sometimes I evaluate studies that they found 20,000 different OTUs in one sample of one horse. And I think, biologically that would not make a lot of sense, because it's very unlikely you have 20,000 different species of bacteria. This would be a very high richness, to an environment that we would expect to find much less or much lower richness. So it makes me think, it makes me go back to material methods and read with more attention what they did in the pipeline, to make sure that everything they did was adequate and they are not reporting something wrong. All right. Dendrograms. This is another way to visualize similarity, okay? And it looks like branches. And the first study I did, I called that a phylogenetic tree. But the concepts of dendrograms and phylogenetics trees are completely different, and therefore they look like the same, but they are completely different analysis. I learned that later from a reviewer telling me, don't call that a phylogenetic tree. But what we can see here is that it's just another way to represent samples. And you can see here, for example, in this study we did with Louis Camus and Christine Theoret, showing that they induced a surgical wound in the limb of horses. And then we evaluated the microbiota over time to see how they would heal. And also we compared the left member, it was bandaged to heal in a closed environment, and the right one was kept open to heal in a different way. And we did compare the microbiota of those limbs, the left and right. And you can see here that in the 4 different horses, it doesn't matter which horse the sample came from. The most important factor influencing the microbiota composition is the type of healing, okay? So all the animals that got a bandage on the limb, they had one type of microbiota, and all the animals that had the wound open to heal, they had a different type of microbiota. And you can see that there is a huge clustering here between bandaged and open healing wounds, okay? Here is another example of a dendrogram in which we evaluated the impact of antibiotics on the fecal microbiota of horses. And what I want to call your attention here is that you can see a branch here that meaning that all those samples had the same type of bacteria, the same composition. And here we are talking about membership. And what you can see from this analysis is that all the samples after 5 days treatment with penicillin, they had the same type of microbiota. So the penicillin was really shaping the composition of that microbiota and making them all similar, all having the same bacteria. And those were collected at different times of the year. It doesn't matter. Whatever animal got penicillin, they will have the same type of bacteria. And the same is true for animals getting TMS. For example, here, all the 7 horses that were treated with TMS, they had the same type of microbiota. Okay, and the same was true for penicillin. So penicillin, ceftriaxone, and TMS, all of the 3 antibiotics changed the microbiota in a distinct way, meaning that depending on the antibiotic you are using, you can have a different effect on the microbiota. You select different types of bacteria. Okay. One other detail here when you are reading studies, microbiota studies, is that when you are looking at this plot, there are 2 different ways or 2 different analyses. You can be looking at the membership or the structure. Okay, both analyses can be represented by dendrograms and by PCOAs. So sometimes you're gonna see 2 different dendrograms or 2 different PCOAs, one referring to the membership and the other one referring to the structure. Now, I'm gonna give it a little pause on the recording because I'm gonna pose you a question. I wanted to think, or I wanted to explain me what's the difference in those 2 analyses when you are looking at a membership and a structure, PCOA or dendrogram, what we are taking into consideration in one analysis that is not taken into consideration in the other analysis. Okay, so the first thing I want to say, like you just answered me, is that membership it takes into account only who is there, which type of bacteria. And like Laura said, the structure PCOA or the structure dendrogram will also take into consideration the relative abundance. Okay. The second question was just looking at a PCOA or a dendrogram, can I tell if it's a membership or a structure? And the answer is no, you cannot. You have to read the legend of the figure, and there you're gonna have saying figure 2A membership, figure 2B structure, okay? And the other question that came up was if you if the dendrogram and the PCOA will normally agree, and yes, they are made from the same data set using different types of calculations, but if they are coming from the same data set, they normally agree. So if you see a clustering here in the dendrogram, you should see a clustering in the PCOA, even though the type of analysis that they use to generate the plots are different. Okay.
So looking for this, if you say to me it's membership, I can say that larvae and fire, they have more bacteria in common than the bacteria that they have. They share bacteria that beauty doesn't have. And if it's a
Absolutely.
structure, I can say that the abundance of the bacteria for larvae and fire are more similar. They are more closer than for beauty, for example. That's the difference.
Yes, and you can that's perfect. And you can also say that all of those 6 horses, they have they have a more similar bacteria when you compare them to Missy. Okay, because and you can say that those 7 here, they have a more similar bacteria when you compare them to Missy at day 1 and Missy at day 14. That's clustering here, and Missy at day 30, because they are with a branch here that shows more similarity. And this is very interesting because looking at this data, you can see that the bacteria from Missy, it didn't change much from day 1 to day 30. They are all of the samples collected from Missy, they are within this branch here that you can see. So day 1, day 14, oops, excuse me. Yeah, day 14, day 30, they are all within this branch here. So for some reason, this horse, she had a more stable or a very stable microbiota that, yes, it was affected by treatment, but it didn't very much from the beginning to the end of the trial. Okay.
And I have a last question.
Yes.
No, you can go for it, and then I'll ask you.
No, I was just going to say that in this study is quite interesting because if you go there and you search for the controls, and we didn't have many controls. For example, I think Daisy was a control. You can see here that her microbiota didn't change after 5 days. It was still clustering with the baseline, meaning that after 5 days, she didn't get antibiotics, but all of the other had. So her microbiota remained stable from day 1 until day 14. So it was, and her day 30, it's here. So it was very good. I was very happy to see results. You can ask your question.
My last question is, what's the difference between the dendrogram and the phylogenetic tree?
So the phylogenetic tree will compare the genetic similarity. Okay, how close to, let's say if we were doing a phylogenetic tree, then we would be comparing the genetic similarity between 2 different bacteria. Let's say here, Lovely and Fire, they would be E. coli and Shigella, because they have a very close genetic genome, and then therefore they would cluster together here, and we say that they are cousins or they are very, very similar, but it's just the conceptual definition is different, but the way they look like is the same.
Thank you.
I'll show you an example. I think the next slide, it's here. So here is a study that they compare the genetic similarity from the great apes, and then they compare the fecal microbiota of each species. And so here is humans, for example, and what they could see is that the composition of the bacteria really follows the genetic similarity. So for example, I believe here are chimpanzees and here are gorillas. So they are, I can't remember, I'm maybe I'm mistaken, but they are very similar to each other, the microbiota. And if you look at the genetic composition, they are also similar to each other and more similar to chimpanzees and more similar to humans, because we are cousins when we are compared to other great apes. So I can't remember which one is which, but if I remember well from this study, one it's a phylogenetic tree and the other one it's a dendrogram, just showing the similarity between them. Okay. And here I wanted to tell, so this is the study of José Antonio Bessegato from Brazil, and what he did was to compare the microbiota of beef cattle arriving to a feedlot unit. And it's very evident here, you can see in green, those were animals at arrival day. So they were having, they were on pasture, and therefore having a forage-based diet. And you can see the similarity, the clustering of this microbiota here. It's very evident. And then the dendrograms are good to show the dynamics of the microbiota. So here it's at arrival, and then in yellow here you can see the shift that this microbiota had 15 days later. After they change the diet, they start getting more carbohydrate-based diet. It completely changes, and you can even see here that this group in red were animals treated with antibiotics, with virginomycin to increase, to enhance growth. And therefore you can see a nice clustering of those animals treated with antibiotics compared to the control group. And as they move on to the end of the fattening period, you can see that the microbiota carries on changing, and this is at the time of slaughter. Okay, so,
Marcel, I have a question.
Yes.
What does mean the different colors? We have two different greens. So what does it mean when we have one?
Yeah. I'm glad you are paying attention. So the light green are the control group, and the dark green are the treatment group. So at this point, there was no difference in treatment, okay? They were all at arrival, but we were already dividing them into, you're gonna get antibiotic, you will not get antibiotic. So I just put here because if I see a clustering at arrival, we could say, okay, the animals treated with antibiotics, they had a different type of bacteria, but this difference was already present at arrival. But that's not the case. You can see that there is a random distribution here. We get like one goal, it was randomly assigned one goal to a treatment, one goal to the control group. And here you can see that there was no difference in treatment groups, between groups at arrival. And after 15 days, you can see a big difference because you can see the clustering here. But these animals carry on receiving antibiotic, and at exit, you're gonna see here that the purple and the red are control and treated animals, and you cannot see a statistical difference anymore. So meaning that when they were leaving the feedlot, the antibiotic was not shaping the microbiota anymore. We think that, I don't know, the rumen bacteria can start metabolizing, get adapted, and starts metabolizing the antibiotic, and then you don't see this effect in feces anymore. And in fact, we analyzed the rumen microbiota of these animals, and you can see that at the slaughter time, there was an impact of the virginomycin on the rumen bacteria, but not in the feces. Okay. All right, so now we move to heat maps, and I used to see more studies using heat maps in the past, but now not so much anymore, but it's still an interesting way to represent the microbiota. And it's a very simple concept. So let's consider we have different points here, and what I want to do is to measure the distance between point A and point B. And it's very clear here that they have the same distance. A and B have the same distance than A and D. So A is a similar distance between those 2 points. But if I compare the distance between A and F, you're gonna see that it has a longer way to arrive there than if I want to go from point 1 to point B. Is that clear to everybody? So let's say I get a ruler and I measure these distances and I'm gonna create a table like that. Let's say this is centimeters, for example. So the distance between A and B is 184 centimeters, but the distance between A and F is 231, okay? And what I'm gonna do here is to assign different scales of colors to the different distance. And let's say I'm gonna say that the shortest distance, I'm gonna call it white. And the longest, the further distance, I'm gonna call it black, okay? And anything that is in between, I'm gonna assign grays or shades of gray. I think there is a movie, no, with Disney. So just to make it clear, if the distance is more close to it, the longest is going to be darker, and if the distance is closer to the shortest, it's going to be lighter. Okay, and this way you can make like a plot like that, and this is the other way around, but it doesn't matter when if you're gonna call the shortest here, you call it black, and the longest you call it white. Okay, so that's what you're gonna end up here with a heat map, and I'm gonna give you some examples. The important thing in heat maps is always the first thing you're gonna do is to search for the legend. Okay, so here you can see that everything that is in blue, it has a relative abundance of 0, and everything that is in red, it has a much higher relative abundance. Okay, so in this example we have two different groups of cows, 1, 2, 3, 4 animals, 1, 2, 3, 4 animals. And these one are getting one type of diet with byproducts of the industry, and this one is getting corn. And you can see that all the cows that are eating corn, they have basically 0 of this bacteria. So the abundance was 0, and 0 of this other one. But they had, okay, so here another example, they had a very low relative abundance of this other genus here, but the cows that were getting distillated grain, they had, or fermented distillated grain, they had 0 of these other species here. Okay, so just looking at that, you can kind of tell relative abundance, and you can kind of tell differences between the two groups. Okay. Another example of humans that had comparing the microbiota of obese versus lean humans, and you can see that people, I can't remember, I think obese people were the ones with high firmicutes. So those here were obese, and if you look at the legend here, you're gonna see that the relative, or this is our values. You don't, not necessarily only relative abundance, but you can look at correlations, for example, and see that they have a different numbers, okay? And therefore, you can see similarities between them, the two groups. And here is very clear that all of them had correlation, different correlations between those individuals. Okay. Here is another one. So let's find a legend. And here we have the different colors. It's again relative abundance at the different phyla. Okay. And here is a legend of the period. So those in blue, here is the time where they were getting colostrum, so newborn calves. And as they get older and older, they change the diet, and you can see changes in the microbiota. Okay, so everything that is red is more than 20% abundance. So you can see here that the Pasteurellacea here is at the family level. Yeah, see, I found a error here. If I was reviewing this paper, I would say, why are you calling phyla relative abundance if you only have families here? Okay, so this is wrong. Okay, but let's say it was right. And here in this family, you can see that they have a very high relative abundance at this point because it's red. But if you move there, you can see that starts to decrease. Okay, so it's just a kind of a heat map that you can see differences over time in the composition of the rumen microbiota of cats. Good.
I have another one question.
Yes, send it to me.
Families, they assess families with biome, is that right?
No, hang on there. We're gonna talk about that in the second half of the class. Okay.
Okay. Thank you.
And I like when students start asking questions of what we are talking ahead, because it means you are thinking ahead. Okay, that's very good when that happens. It's very good sign that you are not sleeping and that
No,
you are
I'm not.
understanding what I'm saying. All right, so, and this again at the beginning was used a lot, mainly because at the beginning we were doing 454 sequencing, which it was very difficult to use, or maybe Sanger, if you like. It was a lot of work and very expensive, so we could not include a lot of samples in our analysis, right? Like now they evaluate the microbiota of hundreds of people and see the clustering, like you see in Rob Knight's plot, PCOA plot. But at the beginning, we were comparing a much smaller data set. And what that shows is that how many types of bacteria are shared between samples, okay? So here we are comparing the microbiota of 4 different, and this is data from my analysis. We used 4 different horses. So yeah, I think it was from my analysis. So we had, those I think were the healthy horses that were not in the hospital, because I evaluated 6 horses, and here I'm only using 4 horses that had a more similar microbiota, healthy horses. And you can see that 123 OTUs are shared between the 4 of them, okay? But here I'm only 40 OTUs are shared between horse 6 and horse 4. Okay, so this is to tell us the core microbiome or microbiota, which is telling us how many bacteria are shared between all of them. Okay. Now I'm going to ask you a question. Why I'm using only 4 horses here and I didn't include the 6 of them, the 6 healthy horses? And it's a very simple answer, which is it becomes just impossible to visualize data there is more than 4 horses. It's going to become a big mess if I include more than 4 samples here. Okay, so right now that we evaluate the microbiota of 10 horses or 20 sometimes, it's just, a huge mess, and you can see it's not very clear. So that's why I never seen this type of analysis anymore, but it's here for you, for your references. Now we're gonna move into enterotypes, which is basically a machine learning algorithm that, instead of looking at the data set and trying to find clusterings, I'm gonna tell the computer, here is my data set. Find a clustering that you think it is present in this analysis, okay? So if I give this to a computer and ask it to do an enterotype analysis, it would look back to me and say, you are really stupid. Huh? It's very clear that you have two different clusterings here. So you have two different enterotypes, okay? But sometimes you want to confirm your impression about the different clusterings that are not very clear, and you can use this type of analysis to tell you. So this is milk microbiota compared from two different farms, and they did, I can't remember if they did, but I'm just giving you an example of a data set that it's very clear that you have two different types of enterotypes. And sometimes we are dealing about milk, so you can call it milkerotypes. I don't know if you can, because enterotypes, it comes, you know, the nomenclature comes from fecal samples or intestinal samples, but it became the name of the analysis. So we use that for different types of samples. In this study, from Nuriyama from France, very nicely done. They were comparing the microbiota of foals, comparing two different methods of weaning. So one method was abrupt weaning. They would, that's how we normally do, just take the foal away from the mare, and they get really stressed. But the other type they were evaluating was gradual weaning. So every day they would take them apart for an extra hour until they gradually adapt to stay away from the mare. And they were hoping that the differences in stress level would also change the reflect on the microbiota, or not hoping, but that was the hypothesis. But what they found instead was that there was no difference between weaning method, but they did find a difference between the microbiota of foals even before weaning, that when they did an enterotype analysis, they found three different types or three different enterotypes. So here C1, C2, and C3. And when they look at the phenotype, when they look at the characteristics of those animals, they could see that everybody that was in C2, they had a very low diversity in the microbiota. They had very high cortisol levels, meaning they got really stressed after weaning. They were producing less short chain fatty acids, especially butyrate, and they had a higher parasite egg load, meaning they were more susceptible to parasite infection. But the other enterotype, they were completely different. And then they can look at the types of bacteria that are present in each of the enterotypes and have an idea about which bacteria is associated with a good phenotype. Okay, so here they found that Lacnospiracia, Prevotella, and Blautia, they were all present in the microbiota of those animals that were not very stressed. Okay. And then just before we take a little break, we're gonna talk about the left analysis, which it's being more and more used. I think I see that in many of the studies now, because it's very easy way to do a statistical analysis to compare differences in relative abundance between groups. So let's say I'm comparing the relative abundance of those two different enterotypes in the previous study, and that's how the left cell would tell me, in this group, Acinetobacter, Bacteroides, and Fibrobacter, they were statistically more abundant compared to this group, and not even more abundant. I'm not going to go into details about the statistical analysis, but it first shows a statistical difference in abundance, and then it does one type of test that predicts if those that are statistically more abundant, if they have any biological importance, if they are what some people call markers for good intestinal health or for whatever you are evaluating for the use of antibiotics. So this type of analysis, it's when we try to find a good, meaningful marker, then you can just upload your data in the website, click a few buttons, and we do that during our workshop. And it's quite easy to do, and you have your results. For example, in this study we did in collecting milk samples from farms, from tanks, in farms in Brazil, in a collaboration study. What we found was that, as you can see here in the membership, there were two different clusters. So one that we did an enterotype analysis, and you can see that we found two clusters. So here was cluster one. The computer told me you have cluster one comprised by those samples, and you have cluster two comprised by those samples here. And you can see that in cluster two, and this is separated by the number of somatic cells in the milk, okay? So if you have higher somatic cell count, meaning that he has kind of a subclinical infection there in the mammary gland of those animals. But here there's a lot of samples that have also high somatic cell count. So you cannot really explain what's driving that separation. Okay, we try to separate that by the time of the year, by the geographic location, by the presence of many different pathogens, and none of what the analysis we did could explain the separation. Okay, so it is still an interesting result. It's under review, but I could do a left set telling the website, Galaxy website, which is the site of the professor from Harvard created the left set, telling it, tell me if there is any difference in the composition of cluster one and cluster two. And what we get is a plot more or less like that, saying, okay, in cluster two, you had a lot of streptococcus, a lot of bacillus that were meaningfully different between those two clusterings. And in cluster one, you had this type of bacteria here. Okay? Is that clear? Because you're gonna see that a lot. So I think it's a very straightforward interpretation of this type of plots. And this is another time when you know you are working too much. I was drinking a beer and I could swear I read left set, and I was like, oh my God, I think
You were working too much.
Again. All right, why don't we take a little pause for a bio break, and then we're gonna start talking about limitations of next generation sequencing studies. Good, so we can carry on. All right, so the next generation sequencing, as you know, it is really good. It's considered a revolution in science because does the analysis of the DNA strands in parallel. Okay, we call that massive sequencing or high throughput sequencing because it really yields a lot of results, a lot of DNA reads. Okay, so it's very good because it's fast, cheap, it's easy. The machines are really compact now. It fits anywhere in your lab. But there are many errors that can be introduced in any of the steps of the analysis. And also we're gonna talk about low taxonomic resolution. And finally, most of the studies we do about microbiota, they are just descriptive. So they doesn't prove anything. They just make associations. And we're going to talk a bit about that. All right. So like I said, any steps of the analysis can introduce error. If you use two different extraction kits, you can have different results. If you use different primers for the same region of the 16S gene, you can have different results. If you use different PCR conditions, you can have different results. And one of the most known and important bias that we have, it's what we call PCR bias, okay? Look at this example here. Let's say we have this environmental sample, or it can be a fecal sample that has a high relative abundance or a high absolute or true abundance of this type of bacteria, but also you have a rare bacteria that is the red one. And what happens after one cycle of PCR is that here we had 5 green bacteria. So when you amplify that, they're going to become 10 different because you're going to double that, right? And you double the rare bacteria too. So the red one, we had 1 and now we have 2. If you run another cycle of the thermocycler, now instead of 10, you have 20 green bacteria. Instead of 2, you have 4. Okay, now imagine that going on and on for 30 or sometimes more, 35 cycles of PCR. And I'm gonna ask you, what's gonna happen to the red bacteria?
It's going to become abundant. Very.
Well, it will become abundant, but what about the green bacteria?
Also become abundant.
Much more, right? So at the end of those cycles, you're gonna have millions and millions of copies of the green bacteria, but not as many of the red one. And when you are going to get this DNA to do your analysis, and you're gonna load the sequencing plate with that DNA, there is a limited amount of DNA strands that can bind into that plate. And therefore, the probability that the DNA from the red bacteria will be lucky enough to attach there, it's much lower, okay? So what you do when you use a lot of PCR cycles, you're probably gonna lose a lot of the rare bacteria during your analysis, okay? And then we have the low taxonomic resolution. As you remember well from your biology classes, all the living organisms, they are classified in this, some people call it tree of life, at different levels, okay? So let's say Laura wanted to send me a Christmas card. Thank you very much, Laura, for sending me a Christmas card. But let's say she forgot to put the number of my house, okay? It is very unlikely that I'm gonna get that letter, that Christmas card. Why? Because there are around 300 houses in my street. Okay, the mailman, he or she not gonna be knocking all of the door and say, hey, is there any Marcio here? Okay, so it's just gonna go back to Laura or end up in the garbage because I was not specific enough. And with the bacteria, it's the same thing. We can compare the different the classification of the bacteria with the address that we need to put it in a letter. And when we do the Illumina sequencing, which is the most used for microbiota studies at the moment, what we have is a good, or not as good, but we can work with a classification at the genus level, okay? Meaning that we don't know which species we are talking about. And this is a big mistake I used to see a lot when I was reviewing papers, and not as often anymore, of people, you know, because you can still classify them at a species level, but this is not reliable. So they were claiming, oh, Clostridium perfringens was associated with, and I said, look, this analysis you are doing is not good to tell it was perfringens. You can still say it was Clostridium. It's not as adequate, but we can live with that, but not perfringens. We don't know if it's perfringens or not, okay? And it should not be capital P here. But we work at the genus level. You can be very confident at the family and higher levels on your classification, okay? Just to put you in perspective, when we are comparing two different phyla, that's the same to compare humans with jellyfish, okay? So if you read a study that they had a very or a statistically difference between phyla, like in the antibiotics study we did, it statistically decreased verrucomicrobia. So it's huge. The changes in that environment is just huge, okay? When you are comparing different, or when you have a change at the family level, it's the same to compare humans and chimpanzees, okay? For example, in that movie, Planet of Apes, when the guy travels in space and time, or I don't know where, he comes back and the planet is taken by apes. That was a change at the family level. So I would say it is still a quite significant change, okay? All right, and here, what I want to say is that, for example, if you take the genome from an E. coli and you stretch that to fit into the diameter of the globe of planet Earth, and then the region we are analyzing with the Illumina sequencing is really the size of a football field, or 6 football fields, sorry. Okay, so this is just to highlight and to illustrate. We are sequencing a very tiny piece of the genome, and we want to classify a whole planet based on this short distance of 6 football fields. Okay, one might argue that, yes, but you know, if you consider that the whole 16S gene is much shorter than that, and that's right, okay, this is true. So, but still, we are sequencing a very small region of the 16S gene. And this is to answer your question, Melanie, that, you know, if you do long read sequencing, you're gonna be evaluating the whole 16S gene, and that's gonna give you a good resolution at the species level. Still, you cannot find mutations and variations that are happening in other parts of the genome, but it's good already enough to classify the bacteria or the bacterium at the species level, okay? But what the Illumina sequencing does is really just a small piece of that whole gene, okay? All right, so now we talk about, still talking about low taxonomic or low resolution of taxonomic classification, and I see that a lot. I just reviewed a paper. They were using primers to evaluate specific primers for two different phyla. Okay, so the singular is phylum, the plural is phyla. They were evaluating two phyla, and my point was, okay, so you are evaluating firmicutes. You found that one group had higher abundance of firmicutes than the other. What does that mean? Why is this important? I don't know which firmicutes are there. There are thousands of firmicutes of bacteria classified as firmicutes. It could be any of them, right? So look at this example here. This study, we compare the microbiota at the different compartments of the horse. So from stomach, passing through small intestine, large intestines, and feces. And what we found is that in the stomach, there was a lot of firmicutes, okay? But also in the distal gut, barely half of the bacteria there were firmicutes, meaning that they had the same type of bacteria, right? In the stomach and in the feces. Is that right? True or false?
Not the same
Sorry.
type of bacteria, the same family of bacteria, but not the same bacteria.
The same family?
This is Microbiota, genus,
Phyla, the same
phyla. Sorry,
phylum.
yes, sorry, the same phyla,
Yeah,
but not the same bacteria. It can be any other. They are just in the same phyla.
Yes, very good. So look at the same analysis at the genus level. Those two families, or sorry, those two genus here, Lactobacillus and Sarcina, they are firmicutes, okay? And therefore, the previous plot, it was showing all of them as firmicutes, even though they have two different types of firmicutes there. But if you look at the fecal or distal gut microbiota, you will see that most of the firmicutes there were Clostridium, okay? So, yes, they belong to the same phylum, but they are completely different bacteria, okay? So every time I see a plot of phylum, and many times I study show, like I'm reviewing a paper and they show the analysis at the phylum level, there was no difference. And I say, so just take this plot from your study and put a plot of the genus level, if there was a difference, even if there was not, because it's much more meaningful. It tells us which bacteria was there, or at least which genus of bacteria, which genera were there. But the phylum level, for me, it's meaningless, okay? Is that clear to everybody?
Yes.
And this, let's see if we have time. I just normally put this here to illustrate, but this is the study of Laura, that she was colonizing chicks with two different types of microbiota. One transplanted from regular chickens, the other one transplanted from organic chickens, and a control group. And what type of plot is this one, Sarah? Sarah Ahvani.
It's a relative abundance. No, it's a
No.
for PCOA.
PCOA, very good, very good. Okay, so what it shows here is that after 7 days they got the bacteria. You can see that there is a clustering of the controls here, and here there is a clustering of the conventional of chicks that received the content of conventional chickens, and here is the microbiota of the donor, okay, from adult chickens that donated the cecal content that we use to colonize the chicks. And you can see that they are very similar, right? And the same here with the organic donors, they are very similar with the microbiota of the chicks that received that content. meaning that the bacteria we gave them stay there. Okay, so this is at day 7. You can see the different clustering there, and then day 14 is the same. Day 28, you know, the control animals start to move along with the conventional group. So you can see the clustering, you can see the dynamics, and here is at day 42. But now the hypothesis we have is that, okay, the organic microbiota has a higher diversity. We believe it's better, okay? And I told Laura, let's make a probiotic or try, at least I want to know which type of bacteria is there. And if we run the Illumina's analysis, you can compare them and you can classify at the genus level, but you're still gonna get a low resolution classification, okay? And I want to know, okay, but which species is there, right? So this is to answer Melanie's question. I can get the same DNA and send that to what we call a long read sequencing. There are the two main platforms that can be used for that now. It's the Nanopore and the PacBio, okay? So you do a screening, you use the Illumina that is cheaper to look at the big picture, and then you select the samples you want based on your analysis, and you say, okay, let's send that for long read sequencing analysis. And this is gonna sequence the whole 16S gene, and it's gonna tell you at the species level which bacteria is there, providing that those bacteria have been cultured, sequenced the whole genome, or at least the 16S, and then they have been deposited into a data bank. And now with the long read, you can go there, compare to the full 16S gene and say, this is 100% matched with a Prevotella, whatever species, or with Clostridium perfringis. Okay, and I can be sure that I'm talking about Clostridium perfringis and not any other Clostridium that might be there. Is that clear? Yes, good. All right, so well, another detail here when we talk about relative abundance is that those are, like the name says, it's relative, not absolute abundance, okay? Let's give you an example that one analysis I got, and this is fictitious, okay? It's just, I made that up. I'm comparing fecal microbiota with a skin microbiota, and this is what I got. So I get many lactococcus, streptococcus, and it's quite obvious this is a milk sample, but don't tell anybody. Just for teaching purpose here. So I'm comparing and looking at this plot, I'm gonna ask you, which sample had a higher load, a higher number of bacteria? Feces or skin? You have to open your microphone.
I don't think we could tell.
Yes.
You don't?
I don't think we could tell with this graph.
Yes, very good. We cannot, okay? We can look at them. They look like they have the same amount of bacteria because both top up to 100%, right? But you cannot say that one sample, fecal sample, let's look at the Escherichia shigella here group, okay? So here we have about 15% abundance, and here we have about the same, like 20%. It doesn't mean there is more Escherichia in the skin than in the feces. There is probably much, much more Escherichia in feces, numerically speaking. Okay, let's say a billion I don't know, a billion, if you count the number of E. coli, you're gonna find a billion here, and it can be only 100 here. But it looks bigger because it's in relation to the other bacteria present in that environment. Okay, even Streptococcus, there can be many more Streptococcus here in this tiny fraction than here, because here we are talking about trillions of bacteria in a gram, and here we are talking about hundreds or a million. Okay, so when you compare relative abundances, you are not comparing the number of bacteria, you are comparing the relation of that bacteria to the others present in that environment. Is that clear?
Mhm.
Good. All right. So what we can do to actually count bacteria and to calculate then absolute abundance. Like I said, you can do direct count that is not viable or feasible, and you can do qPCR using a universal primer that's gonna tell you or give you an idea how many bacteria are there, and then you can make actual proportions about each phylum. And then the other way to count is to use cytometry. Okay. One other limitation is that when you do next generation sequencing, you are sequencing what? DNA, right? You are sequencing DNA. Does that mean that this DNA you are sequencing, it comes from a live bacteria? No,
Not
it comes from
necessarily.
a dead bacteria necessarily, because you're gonna kill it to expose the DNA, right? So even if that bacteria is transient, even if that bacteria is dead, you know, you never know. So you are not proving anything. You are just showing a DNA that is consistent with that bacteria. So it doesn't show viability. And there are different types of tests that you can do to actually show bacteria. For example, in the study we did with Rebecca doing fecal transplantation in horses, what we see or what we know from other species is that many of these bacteria we are giving to those horses, they are already dead because they have been exposed to oxygen, they have been sitting at room temperature, so they die, they just die. And therefore you can still find their DNA there, but it doesn't mean necessarily you are giving live bacteria to that animal. And finally, the descriptive nature of those analyses. Like I said, for example, if you are comparing the microbiota of obese people with lean people or non-obese, and you find a difference, that was the first guess. Okay, so obese people, they have a much higher ratio of firmicutes. Okay, well, what does it mean? Does it mean that firmicutes are causing obesity? No, not at all. It just means that those animals, they have a higher abundance. Why? I don't know why. I cannot tell you why. Okay. For example, when I evaluated horses with diarrhea with healthy horses, and I found higher abundances of protobacteria. Let's say I get one example. I got E. coli. Okay, all the horses with diarrhea, they had a lot of E. coli. Does it mean E. coli is causing diarrhea in those animals? Yes or no?
No.
No.
No. it could be anything. It could be inflammation. They had a very high level of inflammation, and this inflammation is selecting E. coli or is killing the other bacteria and allowing E. coli to overgrow. It doesn't mean it is causing the diarrhea. To prove it's causing the diarrhea, you'd have to do mechanistic studies, or if you want to prove that one type of bacteria is actually causing obesity, you have to get this microbiota or this bacteria, and you have to colonize newborns, and then you have to wait until they grow, and then they develop the phenotype or they develop the disease. Some people call that the Cox postulate. So you are actually proving that this bacteria or this type of microbiota is actually causing the disease or the condition. But you cannot see that in microbiota studies. Okay? You can guess, for example, if we treat horses with antibiotics and you see a spike or a decrease of verrucomicrobia, for example, then you say, okay, compared to baseline, all of the animals treated with antibiotics, they had a decrease of verrucomicrobia. This is an observational study, and it makes sense to say that, okay? But like in comparing healthy horses with diarrheic horses, and you see many horses with fusobacterium, for example, you can argue that this bacterium causes diarrhea, or if it's Clostridium difficile, you can say, yes, it produces a toxin, but, you know, you can discuss that, you can make hypotheses, but you cannot prove. Is that clear? All right, I think we're almost done, yes. So have those basic concepts in mind. Like I said, you're gonna need that to read and to understand those type of studies. And I also said, it's easy to understand, but it's also easy to forget. So get back to the paper of basic concepts, get back to the recordings of this class. If you have questions, just let me know. And then at some point, you're gonna be reading, presenting papers, and in the future, you're gonna be, hopefully, reviewing papers. So it's very important to know the limitations, and when you read a paper, and this is what we're gonna train during this course, okay? You're gonna bring it to me, and I'm gonna ask you questions. I'm gonna challenge you to ask what type of analysis they did and what are the limitations, like, you know, just at the many different aspects of the studies, okay? And then, yeah, when the next phase, when we are discussing your studies, then it's important to have everything all of this in mind to select the right methods and not arrive to the end of your study and realize, oh, I should be using this and not that. So it's very important to have this type of knowledge in advance. Okay. All right, so just give me 1 second to stop the recording.